Module 4, Objective 6, identify the components of the neuromuscular junction, that is the junction between the neuron and the muscle, and summarize the events involved in the neural control of skeletal muscles. The neuromuscular junction is the synapse between a neuron and a skeletal muscle fiber. The axon terminal of the motor neuron releases a neurotransmitter into the synaptic cleft. So there's a little bit of terminology here that you may not be terribly comfortable with, though it's usually covered in psychology. Neurons are cells that have this sort of characteristic shape. Oh, that was poorly drawn. Where you have a regular cell with cell body, and you have a nucleus and all the organelles, but you have these short processes that extend off of the cell. These are called dendrites. And then you tend to have one long process called the axon. And at the end of the axon is the axon terminal, which we saw somewhere on this slide, the axon terminal. And within the axon terminal, we have vesicles that are filled with neurotransmitter molecules. And the neurotransmitter at all neuromuscular junctions is a molecule called acetylcholine. And you'll learn this molecule well, ACH. ACH is going to bind to and open a chemically gated sodium channel on the muscle fiber. When it binds its receptor, sodium is going to enter into the skeletal muscle fiber. It's going to cause a process called depolarization, and you get the formation of an electrical impulse or an action potential. Again, I know there's a lot of terminology. I'm going to repetitively go over this multiple times. So let's look at some images. Sometimes it's easier to visualize. So what we have here in this figure is the synaptic terminal down here in blue. And within this synaptic terminal, we have vesicles that are filled with acetylcholine molecules, these little red spheres. The muscle is located here. This is the muscle. And likely so, we would expect the sarcolemma to be right here, and it is. And we call the sarcolemma that is adjacent, to the synaptic terminal, the end of the axon, we call that the motor end plate. And that term was on the previous slide, and here we see it again. So that is the motor end plate. And on the motor end plate, we're going to have receptors. These are, I'm going to jump to, well, I'll circle it right here. These little purple molecules, these are sodium ion channels. They are sodium ion channels. I'm looking for that terminology anywhere and I don't see it, so I'm just going to have to try to write that. So these are sodium ion channels that bind to acetylcholine, that bind to the neurotransmitter found within the synaptic terminal of the neuron. So, step one, the cytoplasm of the axon terminal is filled with vesicles containing acetylcholine, and acetylcholine is the neurotransmitter. It is going to carry that message from this cell, from the neuron, to the skeletal muscle fiber. So it's going to carry that message, and that message is very simple. That message is to contract. So it's sending the message to contract to the skeletal muscle fiber. All right, step two. A stimulus causes the release of acetylcholine, and you can see here that stimulus is the arrival of an action potential that has traveled down the neuron to the synaptic terminal. Okay, so when the action potential arrives, that's going to initiate the release of the acetylcholine. And that's occurring in step 3. Acetylcholine. These vesicles basically fuse with the plasma membrane of the neuron, and through that fusion, it's going to pretty much dump the contents of the vesicle. So the acetylcholine are then dumped out into a gap that exists between these two cells. This is called the synaptic cleft. It is a physical gap, and that's really why we need the neurotransmitter, because of this gap. Step 4, acetylcholine molecules are going to diffuse across the cleft. Underline, highlight that term diffuse. Remember, diffusion works down concentration gradients. So it is a very random process, but diffusion is going to occur. These acetylcholine molecules are going to diffuse. They're going to bind their receptors, right? Those sodium ion channels. They're going to bind those receptors, and they're going to open the sodium ion channels. And when they're open, sodium floods into the cell. And we can see that happening all along this motor end plate. Sodium is flooding into these cells. And that's going to lead to a depolarization of the skeletal muscle fiber. So this cell is depolarizing as sodium floods in. It's causing a depolarization. So the sudden inrush of sodium ions results in the generation of an action potential in the sarcolemma of the skeletal muscle fiber. And we can see that that action potential is now shooting along the sarcolemma of the skeletal muscle fiber. It's going to be heading out in all directions. At that point, acetylcholine is removed. It has to be removed from the cleft, and that is where the enzyme acetylcholine esterase comes into play. Acetylcholine esterase is an enzyme that is abbreviated ACHE, and it physically breaks down acetylcholine, and we can see acetylcholine being destroyed and broken down here by this sort of moon-shaped enzyme. And acetylcholine esterase is going to return everything back to start, everything back to normal. It gets rid of all the acetylcholine in the cleft. New acetylcholine molecules are in the vesicles, and this system is ready to fire again. So our neurotransmitter is acetylcholine. It travels across the synaptic cleft. It's going to bind its membrane receptor on the sarcolemma. Remember, the motor end plate is the sarcolemma adjacent to the neuron, to the synaptic terminal. It's going to cause sodium ions to rush into the sarcoplasm, and it's then quickly broken down by acetylcholine esterase. The excitation contraction coupling. As the action potential, and I'm going to very crudely draw our skeletal muscle fiber. As the action potential, I'll even change colors, travels down the sarcolemma, it will encounter, let's do green, it was going to encounter the T-tubules. Remember, the T-tubules are just tubes that carry, or that lead deep inside of the skeletal muscle fiber. The electrical impulse, or the action potential, then travels down the T-tubules, where it's going to initiate the release of calcium, which is being stored in the sarcoplasmic reticulum. So as the action potential, and I'll go back to yellow, as the action potential travels along the sarcolemma, It travels down the T-tubules, and it is the arrival of the action potential at the triad. Remember, the triad consists of one T-tubule and two terminal cisternae of the sarcoplasmic reticula. As the action potential reaches the triad, it's going to cause the release of calcium. Calcium is going to be dumped out of the sarcoplasmic reticulum, and it's on its way to binding troponin, which is the next step. So calcium right here is released from the terminal cisternae of the sarcoplasmic reticulum. It binds troponin and causes the troponin-tropomyosin complex to change shape or change position. And remember, tropomyosin is the protein that covers the active site on actin. So as the tropomyosin troponin complex changes position, it's going to expose those active sites on the thin filaments, and the contraction cycle is initiated. If we were to try to summarize all this activity, one would be neural control. A skeletal muscle fiber contracts only when stimulated by a neuron. We call them motor neurons since they control skeletal muscle fibers, or skeletal muscles. This occurs at the neuromuscular junction. The stimulus arrives in the form of an action potential at the axon terminal. Step 2, excitation. The action potential causes the release of acetylcholine into the cleft, which leads to the excitation or the production of an action potential in the sarcolemma of the skeletal muscle fibers. Step 3, this action potential travels along the sarcolemma down the T-tubules, triggering the release of calcium from the triad. Okay, the calcium is being stored in the terminal cisternae of the sarcoplasmic reticulum. 4, contraction cycle begins. The contraction cycle begins when calcium ions bind troponin, resulting in the exposure of the active sites on the thin filaments. Remember, that's actin. This is going to allow cross-bridge formation. Cross-bridge is actin plus myosin. When those two proteins come together, they bind, you get the formation of a cross-bridge. And cross-bridge formation will continue as long as ATP is available. We'll see some other factors that may affect the duration later. Step 5, as the thick and thin filaments interact, the sarcomeres shorten. That's going to pull on the ends of the muscle fibers. It's going to pull them closer together. Step 6, generation of tension. During the contraction, the entire skeletal muscle shortens, producing a pull or tension on the tendons at either end.